Afternoon. Thank you for the opportunity for or giving me the space for presenting this project. As mentioned, I'm a student at the University of Puerto Rico, the Department of Marine Sciences. I'm currently finishing my master's degree in biological oceanography. Um, so today I'll be talking about a project I'm working on titled Know the Invaders, a multi-approach study of semi-soft corals invasions in Puerto Rico. Uh, okay. You see the second slide, right? It's an outline.
Yep.
Perfect. Okay, so I thought it was a good idea to show you a small outline of what I'm gonna be talking about. Firstly, I'm gonna give a brief background of myself so you can get to know me better. Also, I will talk a little bit about the EcoAzul initiative, why invasive species matter, just very briefly. Then I'm gonna be talking about the basics of corals, what they are, and also about what xenids are. Um, After that, I'm gonna be starting to talk about the project itself, which I divided in 4 different parts or chapters, which I'll be explaining one by one during the presentation. Then I'm gonna be talking about the Department of Natural and Environmental Resources Emergency Response Strategy, basically what we have been doing in the reef, and the methods that have been used to control the invasion. And then I'll be concluding the presentation, giving a summary and giving a broader view of the overall project. So now starting with me. um, Apart from being a master student, I'm also a scientific diver. I've been working with coral reefs since 2012, also as a volunteer and then working with different groups. Um, during the same year, I got my open water diver certification. And I've been doing coral reef restoration, monitoring, management, among other things with these different organizations such as HDR Reefscaping. I've done some work with the Department of Natural and Environmental Resources of Puerto Rico, ISER Caribe, Sea Ventures, and other organizations. Um, Apart from that, I'm also a dive master instructor. I provide many specialty courses, and I'm also a technical diver. I normally do cave diving, deep dives using decompression dives, and also I'm a beginner photographer. Um, as mentioned in the beginning, I'm one of the founding member of EcoAzul, which is a new and a emerging initiative. We're just a small group of friends, all from the Department of Marine Sciences, with no prior experience creating an NGO, but we share the passion for the ocean and for giving back to our communities. Our main goal is to grow into a nonprofit organization that bridges scientific experience, management, and local communities to better protect the marine ecosystem. So right now, overall, we're taking our first steps and learning as we go, building connection, and we're again just trying to create something that truly makes a difference. So while we are still in the early stages of the nonprofit, EcoAzul represents a genuine effort to combine all these conservation, education, and community involvement to safeguard and protect our the future of our reefs. So, invasive species come in all shapes and sizes. They impact the ecosystem in many ways. Some notable examples could include the lionfish, tunicates or sea squirts, and seagrass called Alophila stipulacea. It's important to firstly uh, know and identify our invaders, as it is crucial for effective management and mitigation effort. Here you can see just a few ways of impact and possible outcomes that could cause harm to the environment and at the end affect us directly in the economy. Before we start with the main topic, I believe it would be better if we briefly introduce what corals are, just in case we have viewers that don't know what corals are. So corals are colonial animals and which have a symbiosis with a microalgae called Zooxanthellae. Um, they belong to the phylum Cnidaria. They are very relative to jellyfish and anemones. And they come in two different types. Firstly, stony corals or scleractinian corals, which are known to be the reef builders, and also the soft corals, which live within the reef and also provide shelter to other for other organisms. And similar as stony corals, they provide a natural barrier from waves and all other uh, issues from uh, the weather. Now, I mentioned that these corals are colonial, but what are they composed of? So, in simple terms, corals are sac-like animals called coral polyps. Here you can see a picture, a close-up picture or a diagram of them. They have a mouth surrounded by tentacles, and they also have in their tip of their tentacles, they have stinging cells called nematocysts. That's why they belong to the phylum Cnidaria. As I already mentioned, they have a symbiotic relationship with a microalgae called Zooxanthellae, and this Zooxanthellae provides energy through photosynthesis. Um, Stony corals, in general, secrete calcium carbonate skeletons, and also, as already mentioned, they create the coral reefs itself, which is a habitat for the marine ecosystem. They reproduce both sexually and asexually. Asexually through fragmentation and through budding. That's how they, especially these stony corals, that's how they create these massive structures. Now, being a little bit more specific with soft corals, they are also called octocorals, and they are mainly characterized as having 8 tentacles per polyp, and they do not secrete calcium carbonate structures such as the stony corals, so they don't build reefs. They are mainly supported by an internal structure called sclerax, which is also formed in calcium carbonate, but these are very small structures. Now, also briefly, uh, all corals and the whole environment is an important ecosystem because the coral reef provide home to many marine species, protect our coastlines, and also support fisheries, tourism, and in general, local economies. But not all corals behave the same way or provide benefits to the environment, especially those that are invaders. Puerto Rico and the Caribbean have become a hotspot for invasive xeniid soft coral species who have been invading their waters for quite a while, and they are causing drastic impacts, harming the native species and overall helping in the decline of uh, of this marine ecosystem. But what are xenids? So, as mentioned, xenids are soft corals or octocorals, and they belong to the Xenidae family. Most of them are known as pulsing xenia or pulsing soft corals due to their tentacle movement. In this family is composed of over 20 genera, so they are like a small group um, compared to others. Pulsing corals, the pulsing corals I'll be talking about, they are native to the Indo-Pacific and the Red Sea, and they were recently introduced in the Caribbean. They grow and regenerate really fast from even a small fragment or detached polyps, and they are beautiful, and they are used in the aquarium. Uh, Regarding the re their regeneration capabilities, I highly recommend reading Nadir et al. 2023. It's a great paper that sees Xenia umbellata as a novel model organism to study regeneration abilities. It's very, very interesting and a little bit alarming, to be honest. Um, the same way as most of all corals, they reproduce through planula larvae, but mainly they reproduce asexually using fragmentation and budding as one of their methods. Uh, their invasive capabilities could be detrimental, as I already mentioned. They rapidly dominate the already disturbed reef habitats, mainly bare substrate. And at least in the Caribbean, they do not have a natural predator to control their growth. Um, these organisms can easily outcompete stony corals and sponges for space and for light. As these corals, and they are corals, they also need to do the photosynthesis due to their symbiotic algae. We think there is different pathways of introduction. One of them could be the aquarium trade or ballast waters, but also there could be natural ways for them to arrive. Although personally see it very difficult from mainly from where they can come from. Here is a little close-up picture of one of these xenid species. This one is Xenia umbellata. And here's a video I'll try to play so you can see the pulsing movement, which is one of their attractive to the aquarium. I forgot to mention regarding the xenids that their pulsating rate could vary between species. All right, now regarding the Caribbean invasion, I have timeline I always share in my Xenia presentations. But give me 1 second to drink a little bit of water. All right. So the first report of a Xeni species invading the Caribbean was in 2007 in Venezuela with Unomia stolonifera. Then in Brazil, two invaders were affecting their waters. One was Latisimia ningaloensis in 2017, and the other one is thought to be Sarcotilia in 2018. Then in Cuba in 2022, uh, a report uh, of possibly Unomia stolonifera was given, but was recently confirmed not to be Unomia stolonifera, but it is Cynia umbellata. Now, in Puerto Rico, the first report came in 2023, and we also thought that it was uh, Unomia, but no. Later, we confirmed that it was really Cynia umbellata, and later in 2023, we also confirmed another species invading our waters, which is Latisimia ningaloensis. I'll be talking about these two in, in in a few slides during the presentation. So sometimes it is difficult to identify these type of invasive species, these xenid corals, as most of them look very similar. Um, they all share a few similar characteristics. One of them, like I already mentioned, is the pulsing movement. And without accurate identification, managing these species and predicting their impact could become difficult. And that was one of the issues we had in one of our main projects. But first, let me tell you how I got started, how everything started in Puerto Rico, and how I got involved in this. Um, the first report, as mentioned, of these apparent Unomia stolonifera was in 2023. So later that year, I was contacted by Dr. Nilda Jimenez, which is from the Department of um, Natural and Environmental Resources of Puerto Rico, and together with Dr. Hector Ruiz from the group AGR Reefscaping, they invited me to go to the field and confirm if this apparent Unomia species was truly invading our waters, which is the same one invading Venezuela. So the day came in, and while we were on our way to the field, I asked Nilda, like, "Hey, Nilda, is anyone doing any scientific work regarding these xenid invaders?" And she told me that if I'm interested, I could do it. So I started working with the permit, and I got it, and started working with these species using genetics, which is a method I was starting to use in one of my other projects, which is the sea squirts, which are very close relative invert or invertebrate. Uh, they are very close related to us as they belong to the phylum Chordata and share a few interesting characteristics. Um, so I, when I started, I thought it would be fun to use code names to organize the different parts of the project. And I will be explaining each code name in the following slides. I will share the purpose and what I've done in this part of the project, if anything, because some of these are just plans or proposals. Now, starting with codename Aurora, the idea of this part of the study was to identify species and confirm them using genetics and taxonomy. Uh, Now, with this title, I'll be presenting what I did in my first note regarding the xenids, which is already published in the Coral Reefs Journal, and its title is hello. I'm sorry. Can you hear me?
Now we can, but we lost you for a sec. You said,
Yeah, my my headset turned off. I'm sorry. So, uh, I'll be presenting uh, my first note regarding the Senates, which is already published in the Caribbean Reefs Journal. Its title is Shadows over Caribbean Reefs, Occurrence of a New Invasive Soft Coral Species, Xenion velata, in Southwest Puerto Rico. Okay, so our primary objective, which I've been repeating all over the presentation, is to identify the invasive Xenid, uh, which was thought to be Eunomia stolonifera by using genetic barcoding and also taxonomic methods. Out of the different patches we've found growing in the south of Puerto Rico, three were randomly selected, and we carefully collected two samples from each site, preventing obviously fragmentation, as we already knew that they could spread rapidly. We used scuba equipment to go diving and collect these samples, and also to work with them. Um, uh, um, then these samples were placed in a collection tube or Ziploc bags, and when we get when we got back to the lab the same day during the sampling collection, we removed the salt water and we preserved the specimen in a -80 freezer for the genetic process. After DNA extraction, we perform a multi-gene barcoding using 4 markers. And finally, we compare these genetic sequences from our samples with already established xenid sequences that are found in genetic databases to accurately identify the species. Um, Here you can see a map. I forgot to include a bigger map, but I don't know if you can see the purple square to the left side. That would be a part of La Parguera Natural Reserve, and that's the specific site where we collected at least two of the samples at two different sites within that area, and also all the way to the right where it says Ponce, that would be Isla Caja de Muertos, which is an island located in Ponce, and the place where we collected the other sample. Uh, After analysis, our results show that all barcode sequences from the sampled individuals were 100% identical to Cineon velata. So this confirmed the presence of Cineon velata in Puerto Rico and not Eunomia stolonifera. So who is Cineon velata? This organism is endemic to the Red Sea, and we found it growing in both types of substrate, either abiotic or biotic substrates, such as rubble, rocks, sand, sponges, algae, corals, just to say the least. It was growing or overgrowing everywhere, at least in the sites we found them. It is capable of rapid colonization and spreading, which provokes an out competition of native species. So they overgrow their space and take over their resources and impacting the local biodiversity. We've seen Xenia in shallow reefs environment, ranging from 0 to 30 meters, almost 100 feet. But we have also found them growing in the upper mesophotic reefs, so deeper than 100 feet, which is very alarming. It prefers areas with water flow and light availability, but can also grow in crevices, which is something interesting. Um, it prefer, I mean, it can reproduce, as already mentioned. Sorry that I'm repeating myself. It reproduce sexually, but mostly asexually through the methods I already mentioned, especially fragmentation and budding. So this method allows for the population to expand fast and taking over everything. Uh, Overall, the presence of these particular species can alter the reef structure, and we've seen that through the time since 2023, and it's reducing the available substrate and potentially dominating the habitats if not controlled. But wait, there's more. As already mentioned, sorry for the spoiler in the timeline, we have a second invader in our waters called Latisimia ningaloensis, and this is the title of our second note, which was recently accepted for publication just about 4 days ago uh, in the Marine Invasion Journal. Uh, so this species is called Latisimia ningaloensis. Um, so Uh, so when does these other species started? So while we were working with Xenia umbellata, we received these samples from another possible species from Dr. Nilda Jimenez and the department in general of natural environmental resources. And with the images, we thought that it was just a colomorph of Xenia, but no, it had some weird distinction. So we took it and we started working with it. So in this study, also similar as Xenia, we wanted to confirm if this other holomorph or morphotype was a variation of Xenia or just another species. So we employed the same methods as the one in Xenia. So I think I forgot to mention that these samples were obtained from the island of Caja de Muertos. I mean, were received from the island of Caja de Muertos, where we already confirmed the presence of Xenia. So we analyzed their morphology. We went deep into the sclerites taxonomy method, and to be able to differentiate these species and confirm with DNA with the DNA barcoding methods. So our result indeed confirmed that this was another species called the Latisimia ningaloensis. Uh, Similarly as uh, Similarly as Xenia, this species was overgrowing everywhere, but in addition, we saw it growing in seagrass, which is also alarming. And seagrass is protected here in Puerto Rico. And these Latisimia ningaloensis origins, they are endemic to Australia. Um, just to be a bit brief regarding its invasiveness, habitat, and ecological impact, it is truly pretty much the same as Xenia, but it's a little bit less aggressive. We haven't seen them so propagated compared to Xenia. Um, and we after this, we truly think, or at least me personally, I truly think this came from the aquarium trade or maybe ballast waters. Um, these species are very opportunistic. They literally overgrow where there is available substrate, especially now that all our coral reefs ecosystems are being have been declining through the years. So they take over everything and start overgrowing uh, other organisms and the substrate itself. Uh, these plus coral bleaching and diseases is serious business. Our coral reefs are being is very alarming. I truly don't know how to explain it, or I'm trying not to be so drastic, but yeah, it is very, very alarming. Uh, overall, these Seni species are a little bit difficult to identify in situ. Some of them share similar colors, so colors are not truly very good identifiers, although very general. Xenia tends to be light brown to beige, and Latisemia is more bluish gray, but we can also see them in brown tones. That's why I primarily used these DNA barcoding methods and the sclerites method to confirm the species. Xenia is from the Red Sea, and Latisemia is from the Ningaloo Reef in Australia. How did they came to the Caribbean? That's a very good question. Also, another thing that could work as an identifier is their pineal count. It could for Xenia could be between 20 to 30 per site, and it has one to two rows of these pineals. Similarly, in Latisemia is about 17 to 37 per side, and it has in some cases more rows than Xenia. So this is another part of the ongoing project called codename Pathfinder. The main purpose of this part of the project is to identify possible pathways of introduction by assessing the different sites we've found them, and to use genetics to locate their possible origins. Basically, comparing genetics from Puerto Rico and other places around the world. This is just an ongoing project. I still don't have any results on this, but I'm slowly working on it. Uh, so so far, the Department of Natural and Environmental Resources created a map of the scenic patches that have been documented or found. And we found a little over 16 patches, mainly concentrated in La Parguera, but also near Caja de Muerto, near the Caja de Muertos Island, where we only found Xenia and Latisimia. And just to prevent any confusion, at least in La Parguera Natural Reserve, we've only seen Xenia out of all the samples we've obtained and all the patches we've assessed. Another part of the project, which is also an ongoing project, is the codename Omnigene. Uh, all these code names idea is just stuff that come in my mind, and it's just a way to organize myself. It's just a personal thing. I don't know if any of you do any of this in your research, but I like to put funny names and stuff like that, maybe references to movies and such. Anyways, on this ongoing project, the purpose is to understand the xenid invaders and their possible associates, and to identify specific genomic features linked to their invasion. And regarding that, we recently completed a project led by Dr. Alex Veglia, which is also one of the members, founding members of EcoAzul. Uh, and in this project, what the how to explain? The first genomic resource for Xenia umbellata was generated. So this is the first genomic source in the Caribbean. So in the project, about 80% of the Xenia umbellata genome was obtained and compared to presumed Xenia genomes from the aquaria. And we found significant divergence. We also identified one of the associated symbionts, Durusdinium, uh, which is a dinoflagellate that is very resilient. Uh, Dinoflagellates are just macroalgae that live in the environment or as a symbiont to other organisms. This is particularly interesting because it suggests that the coral, this Xenia, may have maintained its original symbiont partner from its native location, arising the possibility that microbes themselves could act could act as hidden invaders, an aspect often overlooked in discussions regarding biological invasions. So with these projects, we're just trying to go deep into this invasion. All right, so this is the final code name, or the last code name, which is I named Nexus. In this project, the idea is to help us understand the mechanisms of invasiveness and the impact that invasive xenids have caused to the ecosystem. This is also in the early stages. Now, to answer the question of what has been done in Puerto Rico regarding the control of the invasion, uh, here in Puerto Rico, since the beginning, after obviously all the permits and all that, the Department of Natural and Environmental Resources from Puerto Rico launched a emergency response strategy that is led by Dr. Nilda Jimenez, where they started using two methods to handle these invasion. One of them would be the manual removal, where colonies were literally chiseled out from the substrate, preventing harm to other organisms. and then they were bagged underwater and then lifted using lift bags. And then when out of the water, we carefully remove it out of the entire marine environment. So far from the beach, very far. And then the group put it, like in a specific area to remove it. Yeah, to just trying to summarize. Another method that was the last one that was being used is the cement cover, where the affected area was literally covered with cement, but we targeted only where the xenia were growing. We were literally being specific to the polyps. So we're not the group was not covering the whole reef. We were going directly to these invasive species. Um, it was proven to be the best, more effective way, as it prevents these invaders to regrow, and it creates a new substrate for other native species. There is also information from the department, and they go to the specifics. Uh,
I think we lost you again.
Sorry, my headset turned off again. Sorry about that. Um, Anyways, this was one of the best ways uh, for handling these invasive species. Now, to conclude, uh, I organized this whole study in all these four codename projects, each with specific focus. Aurora, for example, again, just to you we use taxonomy and DNA barcoding to confirm the identity of the species. Pathfinder to explore the possible ways of introduction. Omnigene to develop genomic resources to better understand these invaders and their associations, and codename Nexus to investigate or to study the mechanisms of invasiveness and their impact directly in the ecosystem. Overall, uh, whoops. Overall, these projects will help us know our invaders to eventually manage the xeniid invasion here in Puerto Rico more effectively and possibly in other locations. Um, but truly, and answering one of the question I already saw in the chat, this project uh, is only one piece of a broader initiative, which personally I would like to follow as my possible PhD um, uh, project. And the idea is to address multiple invasive species in Puerto Rico and the Caribbean. The main goal is to combine biology, genetics, and microbiome research to develop, again, effective removal strategies, uh, prevent future introduction, and overall to protect the balance of the ecosystem and its biodiversity in Caribbean reefs. And with that, I really thank you for your attention. I hope I didn't make you go to sleep. Uh, Here you can see my contact information, and I'm open to questions. I'll do my best to answer them. If I don't have the answer now, we can get in touch later and try to answer those questions.
Thank you
Whew.
so much, Daniel. That was a great presentation, a lot of moving parts, but it was so interesting to hear about all the details of what's going on and all the species. You covered a lot, and we appreciate your time today. So let's see if we have some questions. So far, there is one in the chat, and it says, "Did you make competition or aggression between Xenia and Latissima? Sorry, I don't know how you say it.
Like a
So,
comparison,
um,
comparison?
competition and aggression. Did you see
No,
between the two
n-
species? Is I think what they're asking. If that's not
well,
Tania.
I haven't done a study regarding that yet, but with my observation, we've seen that they are coexisting. Xenia is taking over everything, but Latisimia is also growing uh, with this other species.
I'll ask a follow-up question. Since you said that, it made me wonder, do you think that they could hybridize?
Well, there's always a possibility. I mean, they belong to the same family, so that's a very good question.
We have another question in the comments from Rebecca. Can you suggest ways to preserve a new sample for genetic testing? For example, species ID, symbiotinease, sorry. Maybe I can unmute you and you can ask this. If they are reported in nearby U.S. Virgin Islands, we have permits for mitigation. Rebecca, can I unmute you? And then you can better ask that question since I kind of there.
Sure. Yeah, you went over my question. I was just asking if we don't have access to a -80 storage unit, how can we preserve samples for DNA testing to include symbiotic ACA and species identification? Does formalin like Z-Fix, does that work? Or
Well, at
different?
least with the xenids, I've only either used either ethanol or removed the entire water and put it in a -80, and then analyze the samples pretty much the same day or week. But with my other projects regarding, for example, the sea squirts or tunicates, I've used 90%, 95% ethanol, and I've preserved. They last a while out of the freezer with ethanol, but I highly recommend a freezer or analyze or at least extract the DNA samples the same day. The it is better if the sample is fresh. That's what I truly recommend.
Well, maybe I could impose on the St. Thomas University that does have a -80 freezer to take our samples. I work for a small nonprofit on St. John. One of the tricky things about ethanol, either 90 or 95%, is that to get the DNA suitable type, you can't get it shipped here to the Virgin Islands because it's explosive.
Okay.
It's like methylated or non-methylated, one or the other. But the DNA type, unless you want to send me a little jar by mail.
Yeah, I think we can work it out. Yeah. I
Really?
can contact my professor, Dr. Nikolaos Schizas, which is one of the lab I've been working on, where I have all my samples. So yeah,
Yeah, because
if you
I
can, if
expect that it's going to arrive in the USVI really soon. And although we could just smother it and pluck it and stuff, it would be really good to be able to get the species identified and maybe get you some more information about the
yeah,
spread.
it's very important to confirm, over in general, confirm what species is
Yeah.
invading and make sure they are not native, and if they are truly invading the ecosystem. Uh, I wouldn't recommend just killing random animals, It's better to know what it is.
I know that there are some native soft corals that resemble it, like erythropodium, but are there any pulsing? I mean, if you see the pulsing motion, isn't that
That's
adequate to
one of the
confirm?
main characteristics. They pulse like this, like very much. I don't know if you can see in this presentation, there is to the left, it's small. You can see their
Yeah.
pulsing movement, but that's Cinia umbellata. other species could pulsate slower, and also, especially with the image below, they were a little bit distorted because they are in the lab under a microscope, so they could slow their rate. But yeah.
Yeah. The rate is hard to identify in the ocean where there's currents moving things around, but you can at least see the pulsing action. And I thought that was adequate to confirm that it was an invasive one.
Well,
Not so?
we can confirm that it could be a xeniid. The idea is to confirm if it's truly, for example, at least these two and Unomia that are invading, because it could get confusing, especially with other octocorals that could look very similar, but they are not invaders. They are native corals.
Well, if you get me some ethanol, the denatured type, then I'll let you
Yeah,
identify
please, note my email. You can send me an email on any of those two provided in the presentation, and I'll talk to my professor, and yeah, I believe we can
Thank you.
work,
And I see
do
that
it.
Victoria put something in the chat about getting RNA later or another product too. So thank you.
Okay.
Yes, thanks. And everybody who has questions about the genetics, to make sure you get Daniel's email right there. And good luck, Rebecca, getting your molecular grade ethanol so that you can collect some specimens if they do show up. So yeah, that comment is gone, but hopefully that answers your question. And then we have another question here. I know many coral species, this is from Allie Putnam. I know many coral species are in decline due to climate stressors. Is there any information known on the climate environmental tolerance of these new species? And they have a follow-up question and wondering if the removal was effective at all 16 sites where you did removals.
Okay. Um, can you repeat the first question? And I'm sorry.
Yeah, don't be sorry. So the first question is, since many coral species are in decline due to climate stressors, is there a lot of information known or any information known about the climate or environmental tolerance of these two new invader species?
Uh, I've read that they are very tolerant. I haven't done any study regarding that, but doing a brief research in Google Scholar, for example, you can find some papers regarding all that information. Out of my observations, we've seen them very resistant to temperature and very shallow waters. I forgot to mention that Laticimia ningaloensis that we found only in Isla Caja de Muertos in Ponce, we saw it growing in very shallow waters, under 5 feet, in the summer through all these years. So they are very resistant.
Another
And
great.
also,
Oh, go ahead.
I'm sorry, and also, for example, Xenia, we've seen them from very shallow all the way to the upper mesophotic reef. So yeah, I think they were very resistant. Regarding the second question, I believe the number I've shared was 16, but there are more sites. But we we've only worked, I mean, I've only been either a volunteer or a collaborator in the removal or using these methods, but we only worked with a few. I believe uh, it was at least 5 sites. We either used chisel and hammer to manually remove Xenia, or and also cement, but the cement method was the most effective. Yeah.
Yes, I think that's all in the chat. Does anyone else have another question? Anyone else you can raise your hand if you'd like me to unmute you. If you want to verbally ask your question yourself, if it's a little hard to put in the chat. I will note too, it's interesting. I wonder if this species, rather than ballast water discharge, if this species is an aquarium release, which I've kept them in an aquarium, so they're, I've seen them in aquariums, or it's a really popular aquarium species. So if they are aquarium release, they might even be more tolerant of in harsh environmental conditions just because they were raised in an aquarium settings. Um,
Yeah, there are multiple ways. I have my personal opinion, but uh,
I wonder
to
if the
just
genetics tests could
it
see,
could, it could.
yeah, if you
Yeah,
try to
because
source the
the idea
population.
is to, which is one of the code names, Pathfinder. In that project, the idea is to compare the genetics of multiple areas, not only Puerto Rico, but also, let's say, compare the ones in Cuba, uh, the possible presence uh, of xenia in Venezuela, for example, or even taking samples from the aquarium. I don't know, just ideas. Um, but yeah, I prefer just to say that it might be very difficult for these type of organisms to arrive to a total different air location. I mean, from for example, the Indo-Pacific all the way to the Caribbean might be very difficult, but who knows? Nature is interesting.
Yeah, species have definitely moved from across the world before. I think it's possible, right? Maybe, I guess,
Yeah, yeah.
corals. Um, so I think those are all the questions. If anybody else changes their mind and has a last minute question, just raise your hand and I'll call on you. But I just did want to let all of you know that this is being recorded and I will be posting it on the Southeast Risk YouTube. That can be found on our website. I also invite you to go to our website and sign up, subscribe to our listserv. And so you'll get our mailing lists to see our monthly webinars. Like Daniel here, we have another one. They're all invasive and climate related. We have another one next month and the month after. We run them in fall and spring. And there are other ways to collaborate and meet other folks and see what kind of research is going on in the Southeast, including the Caribbean. Especially any of you island folks, we would love to have you join the network. It's really important to make sure that you're part of the Southeast and that you're represented here, especially those invaders moving up from other areas. Okay, we have one more question really quick. You observe the method of cement is more effective to control. Can you observe these sites for a few weeks for the monitoring? That's
Yeah,
from Maria.
yeah. We all the sites we've worked in are already marked. We have a map. Everything is in GPS, and we have a specific. They have. I'm not in no way. Uh, I'm only involved in working with the removal. I'm not more. I'm not planning anything regarding this. Is That's from the Department of Natural and Environmental Resources of Puerto Rico. But yeah, all sites are information is collected, where we've worked on, what we've done, all that is, we have a record for that.
I was wondering myself, as someone who has worked with biogeographical accounts of invasive species and their spreading, since the Virgin Islands and Florida may be wondering if the species might be coming soon. Do you all put your sites where the species has been located? Do you put them in some kind of database or report them to things like GBIF or some other database that's publicly accessible?
I believe Agra have a map where all these samples have been collected. But I'm not sure if it's updated. I personally, obviously, when I collect my samples or when I receive my samples, I always have the location, the site name, and the GPS location. At least, for example, on these two notes, the one for Xenia and the one for Latisimia, the location are explicit. So you can just go and visit those places.
Okay, great. Anyone else have any other questions before we end here today? Thank you so much, Daniel, for your time and for staying over a few minutes to answer the last minute questions. Again, I'll just encourage you to visit southeastrisc.org, join our listserv, visit our website, find ways that you can collaborate, see all of our great webinars. The link is in the chat there. Thank you all very much, and keep an eye out for this talk on YouTube. You should expect it to be up likely tomorrow. Thank you all very much for your time. Thank you, Daniel,
Thank
and
you.
see you all
Thank you.
soon.